It's This Week in Creationism, episode number 37. I'm your host, Joel Duff, and here I take a look at the headlines in the world of creationism from this past week. This week, just one headline, just one article, one person, Ken Ham. Ken Ham has made a statement that he's made many times before, that he's been corrected on many times before. And we need to talk about this pattern of behavior. And so we're going to see how Ken Ham says he can't see any visible change. Therefore, no evolution is happening. Why does he keep saying that? Let's look at the most recent example of him making this assertion. And then let's talk about why he continues to make these claims. We've got that coming up. Okay, we're back. I am frustrated today. I'm only going to talk about one story this week in Creationism episode. We're just going to look at one article. I saw it and I said, I can't believe we have to talk about this again. And it's frustrating to have to correct the same error over and over and over again. And I'm going to attempt to correct the error in a different way this time. But we're also going to talk about why this repetitive pattern continues to happen. And so you're thinking, what are you talking about, Joel? What's going on? I'm talking about this article right here, seemingly innocent looking article. Ancient mayflies evolved into what? And you know what the answer is going to be. It's going to be, well, they evolved into mayflies. And then he's going to make what he thinks, Ken Ham is going to make what he thinks is a profound point from this, which is not at all profound. And so let's dig into this article, and I will tell you why I'm frustrated and what's going on here. Breaking news. This is sarcasm. It is not breaking news. And Ken Ham doesn't mean that it's breaking news either. He's being sarcastic about this. Ancient 35 million year old mayflies look extremely similar to modern day mayflies. If you don't have anything else to do for your Answers news program, or you don't have an article to published on the Answers in Genesis website that day, just pull out any fossil and say, look, this fossil looks just like something that's alive today. And there's your story. They make it over and over and over again. Okay, that might not seem like very big news. No. It's not very big news. It's not news at all, but not for the same reason that he's going to say. From a biblical standpoint, of course, mayflies look like mayflies. And they aren't 35 million years old anyway. But those who interpret the evidence with an evolutionary worldview, we have to ask, why are so many of the creatures scientists find preserved in amber virtually identical to their modern-day counterparts? Consider, for example, mosquitoes filled with blood that look identical to modern mosquitoes. As I'm reading this here, I just realized there's another problem. Filled with blood. He's saying they're filled with blood. Go watch my other videos on the hemoglobin challenge, and you'll find out they're not filled with blood. So they're not identical to modern day mosquitoes, and the blood is not identical to modern day mosquitoes. Tardigrades that remain tardigrades. Plant mites that look like plant mites. Frogs that look like frogs. Right? You get the idea. It's a pretty simple message. And that's part of the problem. It's a simple message. That's why it's so attractive to young earth creationists and Ken Ham in particular, for them to make this particular argument against evolution. Oh, look, back in the past, so long ago, an organism looked just like it does today. But doesn't evolution mean change? And aren't we told that all things are always changing? And so if they're changing, but we don't see change, checkmate. No evolution. He's saying, if I can't see change with my eyes and something looks the same from the past and the present, then change hasn't occurred. Therefore, no evolution. It's an attractive, simple argument. It's an argument that would be that a politician would love. Okay, and that's exactly what Ken Ham is. He's an expert politician. He's a modern politician. He says what he knows the audience will find attractive. He says what he knows the audience will understand, even if they don't understand. In other words, he comes down to their level of understanding. All right, so what about this amber thing? What's going on here? He's explaining amber briefly. It's a resin that hardens. The creatures inside are preserved as if in a time capsule. Some of these time capsules, in quotes, like, they're not really deep time, right? Just very recently. We see we're buried catastrophically. All right, so there he's telling his audience, oh, look, some of these time capsules, the only way they could have been preserved is under a catastrophic conditions, i.e. the flood. Preserving them. And now several deposits around the world yield striking fossils that can reveal a great deal about the pre-flood world. The pre-flood world, right? So this resin is trapping insects during this chaotic time. But when were those insects alive? They were alive before the flood. And since they were trapped, so therefore we're actually looking at organisms from before the flood. Except that the fossil we're going to talk about, this mayfly, did not live before the flood, according to answers in Genesis. But that doesn't that's not said anywhere in this particular blurb. All right? He's trying to suggest to his audience that you're looking at the pre-flood world, like all the way out almost to the beginning of like when God had created these things. And when you see this mayfly trapped in resin, and it looks just like today's mayflies, you can have greater confidence that God created a mayfly just as a mayfly is, exactly as they are today, and they have not changed. Mayflies are still mayflies. I think I better say another word about the pre-flood world, because I probably won't come back to it. And in case you're wondering what I'm talking about, when I say that Answers in Genesis doesn't believe that this mayfly is a pre-flood mayfly, despite what it looks like right here on the screen, the text of this particular article. He's going to go on to show that, well, right down here it says 35 to 47 million years old from the Eocene layers of rock. You just go to Answers in Genesis website, look up Andrew Snelling, his PhD geologist, and other articles on their website. And what you'll find is they argue that Eocene rocks, layers that are dated to the Eocene, are post-flood rocks. They're rocks that were laid down after the flood in post-flood times, and therefore insects that are trapped in resin that was produced by trees that lived after the flood are post-flood insects. And so therefore these mayflies are not the mayflies from the pre-flood world. They're the mayflies from the post-flood world. That is a minor point compared to a minor problem in this article compared to the major issues. But it illustrates that virtually every line that is written in this article is conveying a misconception, is factually wrong in many cases, or at best is not explaining things well enough for the audience to truly be able to capture the reality of the situation. But these chunks hardened resin can't tell us anything about the evolution. All right. Can't tell us anything about the evolution of anything. Consider this new find of a tiny mayfly stuck in Baltic amber from the Eocene layer. This tiny mayfly, supposedly dated between 35 and 47 million years old, was studied by micro-CT, which allows researchers to produce a 3D image of the insect using X-rays that capture even the tiniest structure down to the details 0.5 micrometers. And what did this reveal? What do you think this revealed? All right, you want to guess? You knew this line was coming, right? You knew this line was coming. You know, it's as predictable as "Were you there?" or some of Ken Ham's other favorite lines. That mayflies have always been mayflies. The similarities are so great that the researchers were able to place it in an already existing genus. It's true. I looked up the original article. They placed this into a particular genus which contains several living mayflies. So, really, the final line here. So a supposed 35 million years has passed and no evolution has occurred. All right. So from this, we can infer what Ken Ham's definition of evolution is, right? His definition of evolution is organisms changing morphologically, right, over time. In other words, morphologically mean, or their external appearance has to change for them to have evolved. But it also tells us that he thinks that organisms must change their morphology in order for that, like evolution demands that organisms change from one thing into another, that's a requirement of evolution. Has he read an evolutionary textbook? Has he attended an evolutionary biology class? Has he actually talked to evolutionary biologists? Are any evolutionary biologists surprised by this finding? Do the original authors of this paper, are they like, wow, this is incredible. A 35 million year old mayfly still looks like a mayfly. This can't be. Are they shocked and amazed? No, they're amazed by the fossil itself. It's so detailed and they get to look at it very closely. And now that and when they look at these details, and there's a lot of fine details here. And when they make all their measurements, they're not astounded by the fact that this organism looks very similar to a couple other species that are alive today. Certainly, they're not exactly the same as any species today, but very similar. I don't know anyone that's surprised by this. Ken Ham acts like this is like absolutely shocking. Like this should be devastating to any evolutionary biologist, that an organism hasn't changed its morphology very much over time. Because he seems to believe, now I don't think he actually believes this. I sure hope he doesn't believe this. He's been told thousands of times that this isn't true. All right? That this isn't something that is surprising to evolutionary biologists and is expected. It's expected that some organisms will look the same over hundreds of millions of years. It's not shocking. He should know that, right? He should absolutely know that. How do I know he should know that? Because I've told him that. I've told him that dozens of times. Now, it very well may be he's never read any of the times that I've told him that, right? And I suspect that he hasn't. But he has a team of people that read stuff, and I know they've read my material. So he has hired people that have not informed him, all right? Or he's chosen not to be informed. It's not just me. It's dozens of other people that have tried to explain to him about the basics of evolutionary biology. And he continues to repeat the same error over and over and over and over again. It's not an accident, all right? It's on purpose. It's a form of communication. He knows this works with his audience. He's a politician. He's not interested in the truth. He just isn't. He is communicating what he knows works with his audience. He's playing on their misconceptions about evolutionary biology. And rather than teaching them, training them, since he presumably has more knowledge than they do, I'm gonna presume that he's not so dumb that he's not figured this out. All right, that he's never heard anyone explain this to him. And if you're wondering what I'm talking about, because I've kind of said what I'm saying, but I'm going to try to demonstrate it with some data in just a moment. I'm going to say that I think he knows about this. Right? He is playing dumb, but he's not actually dumb about this. He knows the truth, but he's not willing to. He's not willing to be fair with his audience. He's willing to pull himself down to their level, all right, their level of ignorance, and play with that ignorance in order to achieve what in his mind is a greater goal, which is he knows that evolution ultimately can't be true because of his interpretation of the Bible. And therefore, it doesn't matter that if he lies about these types of things, right? It's okay to lie to your audience if what you're leading them to is still a greater truth. And isn't that exactly what we have with today's politicians? A complete comfort level with lying to the audience in order to get them to do what you want, to ultimately change things for something that you think is better, but to get there through the act of lying. Okay, now I have to. I've made a lot of bold statements, so I need to back it up a little bit. You might be thinking I need to back it up a lot. Hopefully, I can do that. All right, so you don't want to stress here. So 35 million years has passed, and no evolution has occurred. What does he mean by evolution here? It's clearly just my perception. I stand back and look at an organism. I don't see any changes. Therefore, evolution hasn't happened. My first thing I want to say is there's actually a lot of small changes in these organisms. So it's not that there isn't any difference. At a 10,000 foot level, they look very similar. Get down to the nitty-gritty, and there actually are differences between them. But granted, they're still not so different that you can't place them in what they call a genus. All right. And typically we think of, and I think Ken Ham has the misconception that a genus of one thing is essentially equivalent to the genus of something else in another group of organisms. And that, if you get into systematics, is not true. But I don't blame him for not being able to understand the equivalency issues there and how we define genera and families and so forth, because those are man-made constructions and don't necessarily reflect the overall genetic difference, there's a lot of different ways to measure difference between things, and different groups, we have different rules for those things. So where do I want to go from here? What I want to do is show you that the amount of external morphological change that occurs in an organism can be highly disconnected from the amount of internal physiological and, more importantly, genetic change that occurs in an organism over time. Such that looking at external appearance, some organisms appear to not have changed simply by looking at their morphology, how they operate in this world, like their niche in the environment appears to be the same today as it was in the past. And so from that perspective, change doesn't seem to have occurred. But this doesn't mean that organisms haven't changed, because there are other measures of change, right? And one very important measure, and really the measure that we talk about in an evolutionary biology textbook is really a textbook about population genetics, is how much genetic change has occurred to the organism. And external morphological appearance is not a good indicator of how much internal genetic change has happened in their genome. So genome can be the A's, the T's, the C's, and the G's, right? The original code. It can be the gene order or the structure of the genome. And it can even include new genes, missing genes, rearrangements of those genes, and so forth, right, over time. Those are changes that occur. And here's what I'm quite confident in about these mayflies. I don't have the DNA sequence of this particular mayfly, but we have the DNA sequence of a lot of other mayflies. Mayflies are very, very different from one another. Even species that, if I were to show Ken Ham, here are two species of mayfly. These two species of mayfly, can you tell the difference between them? And I had him look at them. He'd be like, they look the same to me. And I'm going to tell you, these two mayflies right here, you probably can't see that. These two mayflies right here are more different than you are from a mouse with respect to their genetics, right? They're in their genome, how much sequence difference there is between these two species. More difference between a human than between a human and a mouse. right? And yet they look identical, at least from the non-expert's eye. And under electron microscope, there's going to be a whole bunch of differences. But the overall general appearance of the organism, and even generally what that organism is doing in the environment, how it eats, how it lives, how it reproduces, is almost the same between the two. And yet internally, massive differences between them. How can that be? Let's take a look at that. So here's the original article. I'm not going to go through it from Scientific Reports, but this is just if you want to get the gory details of the morphology and see that there are actually morphological differences between this particular mayfly that's preserved in amber that's 35 million years old and today's mayflies of that same genus. This is where you would find that information. What I want to talk about is the genetic difference between mayflies, because I have to back up this pretty darn bold statement I just made about some mayflies being more different from each other than a human and a mouse are, or really any mammal is from another. All right, mayflies are as different as, they're more different. All right. Mayflies in general, there's 3,000 different mayflies, and they're put into a whole bunch of different families. Nonetheless, mayflies are kind of a group that I think somebody like Ken Ham would look at and think like, yeah, they're all just mayflies, right? They're all the same kind of thing. He might even consider them all one kind. At best, maybe a couple different kinds, separately created kinds. So why am I saying this about genetic difference? Let's take a hopefully a fairly easy example here. So, and I just did a short amount of research to figure this out. And that's another thing I'm frustrated about, is that it didn't take me very long to discover how different mayflies are from one another, doing some basic research. And Ken Ham should have been able to do that much research before publishing his little blurb. 6 mitochondrial genomes of mayflies from 3 genera of this one family, right? And they have inversions, translocations of a particular gene in there. All right, we're going to look at mitochondrial genomes just because they're simple. Mitochondrial genomes is just a single 15 to 16,000 base pair circle inside the mitochondrion. The nuclear genome of a mayfly is like huge and would contain a lot of complexity as well, but I want to keep it simple. All I have to do is show you how even a very simple, relatively conserved genome is radically different between mayflies. I only need to go to the nuclear genome because it's going to be even more different than the mitochondrial genome. And so what you're seeing here is this is a phylogeny of a whole bunch of different mayflies. And let me get my pen out. These bars represent the structure of a set of genes. And what you see here is like this particular mayfly down here that was sequenced. They sequenced the whole genetic code of the mitochondrion. It has 3 copies of one of its genes. And then these genes, and it has a gene that this particular species doesn't have at all. All right? So there are large genetic differences in their mitochondria. By the way, if I were to show like all primates and look at the differences in their mitochondrial genomes, all primates have the same genes and the same genetic order, and they're about the same size, right? Your mitochondrial genome and a chimpanzee mitochondrial genome are just would line right up, and there are a few individual A, T, C's, and G's that are different, but really no other significant structural changes or structural differences in our mitochondrial genome. This is an enormous amount of structural rearrangement that's going on between different mayflies, right? The differences between these different mitochondrial genome arrangements of these genes in their genome is equivalent to or more than the genetic differences between all mammals, right? And this is just in mayflies. Now, we'll talk about why that is in a moment, but I want to show you, I looked up, so I just did a really quick search here. I said, I'm going to look up this particular species. This species right here has a code for its mitochondrial genome. That's its accession number, it's called. So I could just go to GenBank, a national database, search for that particular sequence. I took that sequence and I did something called BLAST. And what BLAST does is it compares that sequence to all the other sequences. And then it will give me an alignment of those sequences, showing me how similar they are to other ones, in which case it's going to, the one that should be most similar to it is the one that's most closely related to it, right? Another species that has a very similar sequence. And sure enough, when I do that, I get this particular one here. And that is one of the reasons why in this analysis, they have a common ancestor. These two species are thought to share a common ancestor. They're genetically very similar to one another. Oh, I just used that word very similar, but I haven't quantified the word very, okay? They're very similar to each other relative to the similarities with other mayflies. But what I want to note is I'm going to show you this one as well down here. And so these two are tied by a common ancestor right here. How much difference is this? I mean, I know a lot of people look at phylogeny and like, well, how much difference is that? And the scale bar probably doesn't tell you much, all right? Here's a scale bar here that tells you like how many mutations there are per number of thousands of base pairs. And unless you understand that scale bar really well, it's hard for you to compare this to say like, how similar am I to another human being? Because you can sequence the mitochondrial genome of yourself and somebody else, and you obviously share a common ancestor at some point in the past, but there's going to be differences between you and someone else in their mitochondrial genome. There's going to be an accumulation of some mutations. If I were to draw you and other people, it would look like this. Here's you, here's someone else, here's someone else. On this particular scale bar, you and I and everyone alive today, pretty much, we would hardly be able to even see the differences between ourselves scaled to the same scale. All right, so it's starting to give you some idea that we have some really long branches here, and the length of the branch represents the number or the accumulation of differences between that particular species and whatever you're comparing it to. But how much difference is this really? Let me go here. So this is the species that I looked up right here, complete genome. And you'll see that it's 15,703 base pairs long. So that's the amount of sequence that we're comparing. And if I compare it to its most closely related species that is known, that has been sequenced, it's only 81% similar. All right, so it's 19% of its DNA is different. That's a very significant amount of differences. And then this one down here let's go back here. That is this particular one right there. All right. And that one's only 75% the same, 75%. So if I show you what that looks like, and this isn't the whole thing, I just took a snapshot of a small portion of the alignment between the one that is, actually these are two sister species. Those two species that look the most similar to each other. And so I've compared them. The query is the one I used, and the subject is the sample that came up that's the most similar to it. And each hash mark right up, each hash mark you see here represents where the bases are identical between the two species. And what you'll see is there's a lot of spaces here, all right, where there's a difference. Now, this difference is there's no base pair there, and there is one there. So that's an insertion or a deletion of a base pair. Here's another insertion or deletion of a base pair. Here's an insertion, deletion. Here's an insertion. Here's a deletion. Here's a deletion relative to the query. All right, lots and lots and lots and lots of differences. How many differences? About 2,500 differences over 1,500 base pair. All right, 2,500 differences between these two species that if I held up to Ken Ham and I showed him, here are two adult mayflies of these two species, he would have tremendous difficulty telling these two apart. All right, and he would say they're the same thing, right? Especially if you saw one in a piece of amber and one today, he would say, well, they're the same thing. They haven't changed. The mayfly is still a mayfly. And it doesn't even look like it changed species. And then you look inside and you look at its genome and you see 2,500 differences. And many of these differences do affect the amino acids of different various proteins, and so do affect the organism. Now, you need something to, 2,500. You're like, 25, what is 2,500? Is that a lot or is it not very many? Because I can make it sound impressive just by like saying it's 2,500 changes. Wow, that sounds like that's enormous. Here's a good comparison. How many differences do you think you have from somebody else? Right, in your mitochondrial DNA? At most. All right. Well, there's going to be a couple exceptions, but we're going to say that, you're looking at 50 to 100 differences. 50 to 100. All right, between two individual people. How many differences between you and, say, a Neanderthal? Because we have mitochondrial DNA sequences of Neanderthals. About 200 differences. All right, between any Neanderthal and then any one of us, about 200 differences. How many, now here's where the rubber hits the road, right? How many differences between you and a chimpanzee's mitochondrial genome? It's about the same size genome, has similar genes in it than a mayfly does. How many differences? About 1,400. 1,400 differences. These two mayflies that look almost identical to each other have 2,500 differences. And they have a different arrangement of their genes in their genome, whereas you don't have any differences in terms of your genetic arrangement of your genes in your mitochondrial DNA compared to a chimpanzee. All right. So from a genetic standpoint, from a genomic standpoint, mayflies have experienced a lot more evolution than other organisms have. All right. If Answers in Genesis says that chimpanzees and orangutans and gorillas are all the same kind, that they evolved from common ancestors on the ark. In fact, they all evolved from the same mitochondrial genome. And therefore, all the differences in their mitochondrial genome are the result of mutations that have occurred, right? They have evolved, right, from that mitochondrial genome into the various mitochondrial genomes they have. How different are they? All right, chimps and orangutans, 1,700, 1,800 differences. Chimps and apes, about 2,000 differences between them, right? So they're within a kind that Answers in Genesis talks about. You have less change than has occurred between these two very similar species that morphologically look like they haven't changed, that the average person would have difficulty telling apart, and yet they've experienced more change. And presumably they share a common ancestor. And so therefore, since that time they had a common ancestor, they've experienced all 2,500 of these changes. Now, let's go out and let me go back and remind you, how different is this species, which we were just looking at, and this one down here that was 75% similar. 4,000 differences. Right, that's more differences than between any two. I mean, pick out any two mammals you want to. Right, pick up any mammal you want. Compare their mitochondrial genome. I don't think you're going to find more than 4,000 changes between any of them. And yet they're all clustered together here in two different genera that are considered very similar to each other. And if I showed you pictures of the adult mayflies from these groups, you'd be like, yeah, they're mayflies. Enormous amount of change. Now, what accounts for, and then if you go back here and you go all the way down here and you go all the way out here, how much change do you think that is? Right? An incredible amount of change. Right? I mean, we're talking about a third of their genome being different. Different rearrangement of their genes. They've had, and we're not even talking about the nuclear genome. We're not even talking about the chromosomes. We're not talking about how different the chromosomes are. Different chromosome numbers. different numbers of genes. Some mayflies have, I'm sure, unique genes that no other mayfly has, right? And so to say that that mayfly in that amber has not experienced any evolution is a ridiculous statement. All right? It just is. Right? When any two mayflies are more different than most mammals are different from each other. Right? Enormous amount of internal differences. So what happens is this. Evolutionary theory doesn't say that an organism has to change its morphology over time. It's not a requirement. It says that it can change its morphology over time if that change in morphology provides a fitness advantage to the organism. So you have a mayfly. And by the way, mayflies are related. They're similar to dragonflies, right? And damselflies. And most people know that, dragonflies have been around since, before the Carboniferous. Long time, right? 350 million years, 400 million years maybe. That's a long time. And some of those dragonflies look very similar to today's dragonflies, right? So again, Ken Ham looks at that and says, well, evolution hasn't happened. From the morphological perspective, let's say you're a damsel. Let's say you're a mayfly 300 million years ago, not 35 million years ago, 300 million years ago. And you have adapted to a particular environment. And in that environment, you got a whole bunch of competitors. You have other organisms. But you're super well adapted to how you reproduce, what you eat. That particular food source is still around for 100 million years. And the particular environments you live in are still the same kinds of environments you live in today, right? Why would they change into something else, right? They're really, really well adapted for their environment. If you're really well adapted to your environment, you're not going to change, especially as the world fills up with other organisms. Because what are you going to change into? If you try to change into something else that eats a slightly different food resource or has a different wing size that's not as efficient, you're not going to be very successful. What's the most successful thing to do for many organisms? To not change your morphology, because it's what works. You stick with what works. But here's the problem for all organisms. They can't avoid mutations. They will experience change with respect to their DNA. Every time they copy their DNA, they're making mistakes. So they're adding new mutations all the time. Well, what are they going to do with those mutations? Any mutation that results in a morphological exterior change that would affect their fitness in that environment is going to be selected against. But there's lots of other changes that occur that don't affect that external morphology. And so therefore those mutations get passed on. And so those organisms are accumulating changes inside them over time. And those changes can include rearranging your genome, breaking chromosomes into different arrangements of chromosomes, even adding new genes that do some kind of internal function that you can't necessarily see just on the outside looking at their external morphology. Many of these organisms may be more efficient inside in terms of their physiology and how they break down food and so forth. They may still be adapting in that way that you can't see on the outside. Most of the changes that have occurred to them are gradually internally resculpting every gene, because many of their genes, like in their mitochondrial genome, some of their genes might be 1/3 different than they used to be. And that includes taking this amino acid over here and changing it, but then you replace it, another amino acid on the other side of the protein might change in order to compensate, so you eventually have sort of the same function for that enzyme. And so if you were to take an enzyme that's made by one of these proteins out of one mayfly and compare it to another, what you might find is they might be 15 or 20% different, even at the amino acid level, like the actual structure of the enzyme. But what the enzyme does in terms of its action might be the same between the two. In other words, you might look at it, that's almost like morphology itself, right? You look at the two enzymes and you measure, like, what are they doing? Well, they're both metabolizing this particular thing at this particular rate. And it's about the same between the two species. And you say, like, well, look, nothing's changed. They're still doing the same thing. Well, yeah, but they actually did change, right? The protein itself has morphed itself over time in both of those organisms separately because they've accumulated different mutations. And so they have two different ways of making an enzyme that has the same function. And in some cases, especially when you get to looking at organisms that are 500 million years old, you can look at the enzymes they have and the functions they're performing. And you can see, like, okay, they're both able to metabolize this, but how are they doing it? They're doing it with two enzymes that, I mean, if you compared their DNA sequence and their protein sequence, they look really different. And you're like amazed, like, how can they even be doing the same thing? But they managed to fold into a structure that's very similar, even though they're made out of different amino acids. And so a protein person would look at them and say, these have evolved. They've changed enormously over time. But from a functional standpoint, they haven't really changed very much. That's because of the power of natural selection, right? The power of natural selection is that natural selection will continue to always preserve the function of that particular enzyme. Because, like, we have to continue performing this particular function in order to, as a whole organism, survive. But you can dink and play with all the different parts of that protein as long as you don't change the function a whole bunch. And so over time, essentially, you replace most of the parts of something and you build it into something else. It's kind of like, you're on a boat and you're going across the ocean. And as long as you don't mess around with punch a hole in the hull and sink yourself, you could remodel the entire boat. And by the time you got to the other side of the ocean, some of you look at it, you're like, man, you started out with this kind of boat, and now you're in a yacht. It's like, in that case, the morphology looks like it's changed a lot, but the core structure, is still the functional unit is still the same. At the end of the day, it still has to float, and that's what enzymes are doing. All right, so I can't stress enough how incredibly different these mayflies are, like from a core molecular biology genetic level. The 3,000 different mayflies that are alive today that are represented in the various families that are here in this picture and this particular phylogeny. This might as well be a phylogeny of all vertebrates, right? The differences are like at that level. The differences between dinosaurs, birds, mammals, and amphibians, right? That's the kind of level of change that these things have experienced over time. And yet they haven't really changed in terms of their overall, like, they're still, like Ken Ham says, they're still mayflies. But that's because the environment has said mayflies are a great thing. They work really well. And so mayflies are going to continue to stay mayflies. I would expect that you come back in 30 million years, I'll bet there's still some mayflies around. And they're going to look pretty similar to they do today. So when Ken Ham keeps harping on, oh, look, you found this tardigrade that's 30 million years old. You found this that's that old, and it looks just like something today. The key word there is it looks like it is the same. It is not the same organism. It's experienced a massive amount of change. Doubtful that they could even reproduce with one another because their genetics would be so different from each other that they would be incompatible if you tried to share a sperm and an egg from a living version today and an extinct version, and you tried to get them to pair and you tried to get their genetics to figure out how to make a mayfly, probably completely incompatible. After all, almost all these mayflies alive today, even species in the same genus, are incompatible with each other. That tells you there are genetic differences between them. So I hear Ken Ham constantly talk about this this just really simplistic idea that just because I can't see something different on the exterior, somehow no evolution has occurred. And so, I guess what I'm saying, it may sound like a mixed message. I'm saying evolutionists don't expect to see changes, except we do expect to see changes. I would certainly expect any organism that was 30 million years old or 30 million years separated from its ancestor to exhibit a lot of change. But where would you find that change? You could find the change in external features. That's very possible. But you also could have an organism that doesn't experience any discernible differences in its external features, and yet would have tremendous differences in its internal genetics. Any organism that's 30 million years old or 30 million separate from another organism is going to have different genetics. I think that's something that evolutionary biologists would predict and expect to see in all organisms. Okay, one other, I just got a couple other things here. This is just another paper that I looked at with another set of genomes. But what I liked in this particular paper was there was a figure, and this figure included the species I just was talking about right here. And so I was just talking about the differences between, I think, this one and this one right here being 75% difference. And this picture here represents, I don't remember which exact species this represents, but all the members of this particular group, which is a family, essentially have this same body plan, same appearance, right? I think Ken Ham would look at all the members of this group and say, like, yeah, they're all the same thing, right? They're the same kind of thing. And not to beat a dead horse, but I'm going to say it again, the differences between these organisms in this group at a genetic level, at the genomic level, of the level of A, T, C's, and G's, the information that's in those organisms is more different, right, than human beings are from chimpanzees and other primates. It's probably more different as you are from a mouse, right? If you just say, like, and young Earth creationists like to talk about, they like to do their stats too, right? About genomes and statistical differences. Oh, there's this much difference between them. Yeah, there's a huge amount of difference between insects and their relatives. Now, I'm trying to think if I feel like I had a lot of other things to say there, but I think that's the main point. Now I've got something else to say about just these types of articles and my frustration with Ken Ham. So this is just a short blurb. It's basically an advertisement for Answers News. They say, like, hey, we talked about this story, or my employees talked about this story. Thanks for stopping by. Thanks for praying, Ken. And then there's this byline, which is on the bottom of most of his blog posts now. Goes something like this. This item was written with the assistance of AIG's research team. So now you learn that what we just read from Ken Ham, it wasn't actually written by Ken Ham. This was written by his research team. And it is not his writing team, he calls it his research team. And he does employ, right, a PhD geneticist. He employs a PhD geologist. Where are they and what are they doing? How do they allow Ken Ham to be so misinformed? Now, of course, I don't really think he's misinformed. As I already told you, I think he's just a politician, right? And he doesn't care at all, about the facts here and trying to actually understand these differences between genetic difference and morphological difference. He doesn't really want to understand or care to understand what expectations are of evolutionary biologists and paleontologists. As I said before, I look around and I say, what paleontologist out there is like scratching their head and like wringing their hands over, like, what are we going to do about the fact that these organisms look the same, like dragonflies look the same from 400 million years ago to today, because evolutionary theory says they have to have changed into something else. They can't still be dragonflies because all organisms are destined to change into another kind of organism? Nobody is, because that's not what the expectation is. The expectation is it's possible for some organisms to change and adapt some of their features such that we would identify them later as being a different kind of organism. But it's not a requirement. It's not an expectation that every organism would do that. As I said before, it's almost an expectation that some would not change over time. It's like, yeah, of course. Yeah, I look in this amber and I'm expecting to find things that are really similar to what we see today. And that tells us something about the stability of certain characteristics. It's really interesting from an evolutionary standpoint to ask what characteristics don't change over time and what characteristics do change over time. Because the characteristics that don't change over time tell us that there is strong selection for maintaining a particular characteristic. That characteristic is fundamentally important for the survival of that type of organism, and they're just not going to play around with it, right? Yeah, they're going to have mutations, and mutations could change, are changing those features. But any of those changes are negative changes and therefore swept away by natural selection and prevented from prospering in future generations. And so finding characters that are maintained for long periods of time in the geological record are important sources of information for identifying the critical features of organisms and the critical functions of those features. So back to this item written by the research team. I mean, it didn't take me very long to do this analysis, and I've just done a really simple comparison of mayflies to another, looked up a couple papers, and quickly found out that mayflies are really different from each other, and even if they look the same, they're very, very different in their internal genetics. And are they unaware of this? I don't know. I don't know if it is intentional ignorance. But as I said before, I've written about this. I've said this. I know that many critics, many Christians who are critics of young earth creationism have pointed this out to them, that this is not a fair assessment. And a fair criticism, right, of evolutionary theory. And yet they continue to make these claims. That's what's so frustrating. I guess what's frustrating is I know that what I'm saying here isn't going to do any good. I mean, it won't possibly make a difference to Ken Ham. And in fact, it just emboldens people like that. It's kind of like politicians. The more you call them a liar, the more they just dig in. And they say the same thing and they double down, right? I mean, that's the word today. It's like everybody just doubles down on whatever mistake they made, even if it was an innocent mistake and they could easily apologize for it and say, oh, I'm sorry, I was wrong about that. No, we just double down and we accuse those who suggest somebody made an error to be out to get them. You know, we question their motives. We call them names. All that stuff. And Ken Ham's pretty good at doing that. He's an expert at doing it. I mean, he's been doing it since, politicians have always been doing that, but he's gotten, let's just say, better and better at it over time. Last slide. This is from the top of the blog post. I didn't show it to you earlier. 35 million year old mayfly looks astonishingly like a mayfly. Again, like I said, whoopee-doo. Doesn't matter. It's not a surprise. By Ken Ham, but as we saw, it's not actually by Ken Ham. It's by his research team that writes his stuff for him. So they're responsible for these errors, but ultimately Ken Ham's putting his name on it. He should be upset with his employees for not informing him. I mean, he should be upset with his employees for not, writing him better stuff. Okay. This is a minor quibble, just to end this off with. But the article that he links to and he sends his audience to, as if anyone in his audience is actually going to click on the links, but if they would go to this site, IFL Science, to read this article. I wonder if Ken Ham knows that IFL stands for I effin' like science, right? That's an acronym for. And I don't particularly care for this particular site. I don't use this site as my science source, mostly just because I don't like that particular attitude, in projection. I'm sure that Ken Ham wouldn't appreciate that kind of language either. So I suspect he doesn't know, or maybe he doesn't care. I don't know. Who can read the mind of Ken Ham? I certainly can't. Yeah, I can only shake my head today. I'm kind of tired of beating my head against a brick wall. Yeah. One other thing I guess I'll mention. I remembered. Here's something I forgot to say. Another analogy. Ken Ham looks at dogs, right? Dog breeds. Likes to talk about dog breeds. Likes to talk about how different they look, and yet they're the same species, right? And that they're the same thing as a wolf. And so he likes to talk about how all canines are the same kind, and they could have changed and evolved. And so he's like, look at all these differences. And I think he's caught up in those, like, look at all the variation that can occur within a kind. God has given these organisms the ability to adapt and change in different environments and become highly variable. Now, of course, he would say they're limited. They can't change into something that's not a canine. But look, they can change into Chihuahuas and Great Danes and stuff like that, right? Huge differences between them. So he's caught up in the just the eyesight thing, and like just using my eyes, right? Instead of saying there's a lot of other information available, and you have to understand the whole package of what's going on. It's part of evolutionary biology. Anyone who learns about evolutionary biology. Let me just note that domesticated dogs, even though they morphologically to someone's eyes look really different, like, look at how much change has happened. They're 99.999% the same. Like, internally, their mitochondrial genomes are all virtually identical. Same sequence, same gene order. If you go into their genomes, right, their nuclear genomes, they're going to be 99.999% the same sequence. It's just a couple individual mutations to important developmental genes and a few variants in like coat color and things like that make all the differences externally. So it's very deceptive. They're basically clones of one another. Domesticated dogs are virtually clones with small changes that make huge external differences. Compare that to the mayfly. The mayfly has an external appearance that looks like it isn't any different. And yet internally, they're vastly different from one another, right? As different mammals are from one another. In Ken Ham's world of understanding genetic differences with respect to kinds of mammals, the mayflies, like virtually every species, should be a different kind in his world, even though they look very similar to one another. Right? So he does not have a holistic understanding of speciation, genetic change versus morphological change at all. All right, let's quit there. Hey, like, subscribe, all those things I'm supposed to say that I always forget to say. And we'll talk to you later. Bye-bye.